Hello, dearest conference attendees. We are about to watch a presentation on strong germline engineering.
Yes.
Welcome, Max Berry.
Thank you. Welcome, everyone. All right, well, we'll just get started. You guys know basically what this is going to be about. So, first off, I'd like to claim the term designer babies back from all the embryo selection companies. We already lost the term biohacking to people who wear smartwatches, so I'm not letting designer babies get turned into whatever people think it has to be. So, obviously, I imagine most people here know, how embryo selection first started. If you got, you're a carrier for Tay-Sachs, so is your partner, you might have a baby with it. You can do IVF, find an embryo that's at least a carrier and possibly just doesn't have the disease at all, not even a carrier for it. And then it started to get used for a few more things. People can use it for sex selection. I'm not going to read that subcaption on that one. And then it moved into polygenic scoring, right? The 23andMe for embryos. Fun fact, I know everyone loves to talk about Gattaca in this field, but Gattaca was actually only selection. There was no editing in there. It's in the script. You can watch it again. Maybe we can do a group watch or something here. So for that, you can select against negative health traits that might be a problem if you're Ethan Hawke. But the interesting stuff that people want to do, right, is maybe you get a taller baby, or what 99% of people, at least in this room, want to do is have a smarter baby, or things along those lines. And that's all fine. It's great. You should do that too. And you can take these embryos and do some more stuff with them. But again, not designer babies. I'm claiming the term, or reclaiming it. And then there was, of course, good old JK. I'm not going to try and pronounce his name correctly, but he's JK. So you guys remember him back in 2018. He microinjected some CRISPR into 3 embryos, and it sort of worked. And CRISPR, I'm going to rant about this a little bit more on this slide and the next slide, but it's really not suitable for human use, and there's several reasons for that. I have a couple church quotes in here, but that's the one I like. He calls it genome vandalism. And it's really only good if you want to just knock out a gene. The way CRISPR works is just cuts and cuts and cuts until the cell just kind of gives up trying to repair, and you get a mutation that hopefully knocks out the gene so it stops functioning. So maybe he does really care about HIV transmission, even though the parents, you can give them antiretrovirals, they're not really going to pass on the HIV to their kid anyway. But it's mostly because there's only 2 or 3 genes that you can usefully just knock out. You need most of your genes. They're important. They do stuff. CCR5 is one that you can knock out and you can't catch HIV. There's some other stuff that it might be involved with, cognitive things or dengue fever, but who gives a shit? There's that one, and then there's PCSK9 for lowering cholesterol, which would also be a good one to knock down, but there's RNA interference drugs that can just do that now, and you take them once a year, and it does basically the same thing. And then there's myostatin. Don't really want to talk about myostatin, but this is not the way to get jacked, or having your baby get jacked, I guess. So there's a lot of problems with this. These are the main ones, is sperm goes into the egg, or just before the sperm goes in, you inject a little bit of CRISPR, and it hopefully does the edit that you want, and then stops completely and doesn't do anything else, doesn't get bored and start chopping other things up. Like I say, knockouts are the only thing you can do reliably if you want to change a letter, if you're interested in editing a bunch of SNPs for certain genes. This is not really the way you want to do it. And there are unavoidable risks that you really cannot handle with either off-target mutations or mosaicism, which is only some of the cells get modified. And it's not great. So here's the ideal case on the left here, right, is you inject the CRISPR and it does the edits, and then the cell, 1 cell becomes 2 and then 4. I'm going to be saying that a bit. And all the cells are modified and they're all perfect and there's no fuck ups. And you take 1 or 2 of the cells out and you sequence them and you go, okay, it looks good. But the problem with this stuff is you can shoot the CRISPR in there and then, the cell divided before the edits happened, and only one of those cells got the edit, the other one just doesn't. And when you have an embryo, you want to presumably turn it into a baby, so you can only take a few cells out of it before it becomes non-viable. So you're taking a few out, and you're just there's some statistical probability there on whether you can get a full kind of accounting of the genome, but you might get unlucky, pull those out, and you get some cells that are modified, and it's well, I guess we're good, and then only half of the cells are modified, and then the kid still gets HIV or whatever else you're trying to do. And the other corollary of that is, right, it does the cutting in the cell, and then some of it in a different cell goes and cuts some essential gene or something you don't want it to cut, and again you happen not to sequence one of those bad cells, and you're like, oh well, I guess it's good. And you can do this, you can do it 1,000 times in embryos, and you can kind of destructively sequence the whole things and get statistical probabilities that it'll be okay. And if it's 99.9%, that's still not really good enough to make a human baby with. So that, along with the other problems on the editing capacity, it's just not something you want to do for your children or other people's children. So is there a better way to do it? Yes. So that previous stuff was the kind of state of the art, and people are still pushing the CRISPR microinjection, but turns out you can just grow embryonic stem cells in culture, and it's called embryonic stem cell culture. So you get the embryo at an early stage, you kind of crack it open, you put the cells in a dish, and then you grow them, and these cells like to divide. They divide about once every 20 hours. So you start with a few cells, and in a few days or weeks you end up with thousands of them. And then at that point you have cells in a dish, which are the kind of ideal case for editing. You don't have an immune system trying to fuck with you and various other things, skin and whatnot. You can just kind of sprinkle in whatever edits you want to do. And that's much higher efficiency, much more reliable. But importantly, so I'll take you through the process. You get a bunch of these cells, say a few thousand just for this demonstration. You hit them with some CRISPR or something. You give them a few days to recover. They'll get the edits that they're going to get. They'll get the off targets that they might happen to get. And then you kind of shake them up and you scatter them on a plate. And I know this is a plate with E. coli for the biologists out there, but it gets the point across. This is some blue-white screening, which is very fun. So, a cell lands somewhere on a dish away from other cells, and then it does what cells do. All the cell wants to do is become two cells. So it divides and divides, and you end up with a little kind of colony. And all these cells in this colony are derived from the same starting cell, so they all have the same genome, which is very important. You want to find a colony. You just kind of start searching through them. You scrape off half the cells, and you send them off for genome sequencing. And it's actually better than regular embryo sequencing. There you have to kind of pull a few cells out, and you're kind of inferring a lot of the genome, like cribbing off of the parent's reference genome when you do that, which is not great. But if you get just a big blob of cells, you can get a fairly high quality genome out of it. And you just kind of start searching through, and you look for a colony that has all the edits you wanted to make, and there's no very bad stuff that you can't just brush off. And from this point, you have a bunch of these cells, and they are embryonic stem cells with modifications and no off targets. And from there, you can theoretically go back and do that again and repeat the process up to a certain point, which I won't get into, but at least a few rounds of this. And that's pretty cool. So now you have a bunch of modified embryonic stem cells sitting in a dish. And I'll get into more of the details on that, but skipping over that, you have to turn them back into an embryo because you kind of fucked up the original embryo. So there's a few options there. Everyone's familiar with Dolly the sheep back in the 90s, and then there's been various pet cloning companies. Don't look in their garbage bins. You're going to find a lot of horrifying looking puppies. But the problem with that, with Dolly and with the pet cloning and people trying to do it in humans normally, is you're taking some skin cell, or if you're a bit smarter about it, some kind of stem cell thing, something that's a bit less aged, and you kind of shove it into an egg, and you're just hoping that egg can, within one cell division, reprogram this entire nucleus into something that's good enough to turn into an animal or a human. And honestly, it's a miracle it works at all doing it that way. There's a lot of really weird, Dolly had all kinds of fucked up organ failures, and yeah, it's, again, not something you want to be doing in humans, at least if anyone's going to find out about it. And I mean, just in general, for ethical reasons, you don't want to do it. But when you're starting with an embryonic stem cell, it's already 95, 99% of the way to this kind of totipotent state. So we expect the efficiency on this should be much higher. Instead of 1% of the cells becoming an embryo and being born, it's a bit higher than that. So you need some eggs for this, but it's not too bad. We just need to see if it works. No one's obviously done it in humans, because then people start asking questions. The second way is a technique called VelociMouse, because we mainly use it in mice. You get some other embryo, doesn't have to be yours, that part doesn't really matter. And it gets up to about the 4 or the 8 cell stage, and you take some of these embryonic stem cells from earlier, and you just kind of shoot them in there. And for, I'll just say, biology reasons, the cells that are already there go, oh, these cells are slightly further along in the developmental process. They're going to form the inner cell mass. I guess that means we have to form the placenta. So they just do that. And then all the baby parts, not the placenta parts, are formed from these embryonic stem cells that you injected with the edits. And it's not 100%, but people haven't really tried to optimize it in mice because you can just make more mice. You don't really need to get it 100%, but there are ways of optimizing that. You can just shoot them in there. That's option 2. Option 3 is the synthetic embryos from Hannah Lab, and I forget her name, but she kind of rips off the Hannah Lab, that other lab, Magdalena, whatever. And they spun that out into a, no shade on her, she also has interesting results, but it's mostly Hannah. And then he kind of spun off Renewal Bio, who are looking into this for absolutely not human gene editing stuff. It's probably, organogenesis or something. But you kind of trick some of the ESCs into forming all the bits, and then you kind of shove them to the bottom of a tube, and it forms an embryo. That's a bit further out, give it 5-ish years maybe, and that could be an option. But like I say, we have some options here. So obviously it's not quite this simple. There's a lot that goes into this. This kind of stuff can routinely be done. I know this meme doesn't quite match for this, but I couldn't find a better one for it, and it's still fun. So mouse embryonic stem cells, I won't get into the whole naive versus prime thing. There's kind of 2 states of embryonic stem cells. They start in the body as naive, and then they kind of get primed, and there's different kind of morphology of them, and they have different characteristics. But mouse ESCs, people just threw them in. They were growing mouse ESCs, and it was like, oh, they're naive and they're fine. You can do the Velocimouse stuff very easily, and whatever else you want to do with them. Humans, it's taken a little bit longer, but thanks to some work from the Serrano lab and Sergey, who is, I think, talking on Thursday about super socks, it's become a bit more promising. There's issues. I keep saying I won't get into this, and then I do get into it, but there's methylation, something called imprinting, if any of you are familiar with imprinting disorders. Most of your genes, they're either like silenced by methylation, the DNA gets little methyl groups attached to it, or it's not. Except in the early embryo where a few very critical genes, for reasons that escape us, but I'm sure nature has a plan, you'll get some where the dad's copy of the gene is methylated and the mom's is not, or vice versa. And the usual way of growing embryonic stem cells in a dish is you kind of sandblast all of the methylation off of them, and then they look like embryonic stem cells, but if you tried to make a baby out of them, you would have all the imprinting disorders known to us, and probably a few novel ones, which could be fun. And that's been the problem up until very recently, and made this basically impossible. But yes, the Serrano lab figured out a cocktail that has not been fully tested with this, because right after they published this, unrelatedly, they all got hired by Altos Labs to work on longevity stuff, which I guess good for them. But no one's really picked up that research. Sergey, I think he's still looking for funding. If anyone's interested in that, it's super cool stuff. He's a fun guy. But I think with this, with the equivalent of a few papers worth of academic research, but done in industry lab, because it'll have to be industry for reasons I'll get into, you could basically get it to the point where you could get this sorted out. Obviously, there'll be a lot of testing involved, and genome sequencing and verification and optimization, but that's just kind of biology. So, speaking of that, it's the next slide. So yeah, academia, that's where most of this work has come from, because obviously germline editing in humans is illegal for now, we'll say, and industry doesn't really want to touch stuff if there's not a chance of return on investment. So academia, they can claim it's to understand the early stages of human development and whatnot. So they kind of get up to these points where it's like, oh, that seems interesting, but if you want to be like, okay, we're going to test this out up to the point where you'd be making a edited baby, they have to answer to IRBs and various other people, their colleagues. So it's not great, and pretty much all of them take federal funding, which thanks to, was it DickieWiki or whatever the...
DickieWiki.
Thank you, yes. That stuff I won't get into. Any creation or destruction of embryos, if they're taking my tax dollars as a God-fearing Catholic, then I won't stand for that. So that's definitely limited them. You kind of need not like a ton of embryos to test this out, but a few hundred at least to get what I would consider to be lab validated. So industry doesn't really give a shit. It's kind of annoying to buy tons of embryos legally for now. Even if they have like Tay-Sachs or whatever and no one wants to implant them, there's probably hundreds of thousands of millions of these embryos sitting there, and a lot of people that just don't want to toss them out, or legally they can't, or whatever, and just kind of stuck with them. But that doesn't matter. We could fix the Tay-Sachs and then also do some edits to them, and run them through this whole process, make sure they're doing okay. Like the FDA appropriations bill, whatever the hell that we're hopefully getting rid of, please. The FDA cannot consider for approval basically a process of implanting a modified embryo, or they can't spend money investigating it, and I doubt they'd work for free. So technically it would be, I mean, no one's ever tried, so we don't know what they'd be charged with if they did try it. But everything up to that point, getting embryonic stem cells and growing them, modifying them, and testing out, the nuclear transfer or the Velocimouse thing or the synthetic embryos, you can do that and grow them up to, there's the 14-day limit, which I think most people have at least heard of. You can grow them a little further than that and see how they look with the methylation and general development kind of stuff. You can't really grow them much longer than that anyway, because they need a uterus, really. But it'd be pretty good as we're going to get without putting them into a uterus, basically. So, like I say, it's just been a matter of funding, convincing people who are interested in this that they can get a return on their investment and not be flayed alive in the media, which I think they stopped caring about that latter bit as much, and hopefully the former will be taken care of soon as well. So general slide on the pricing. I'm not going to say how much the whole program might cost, but I give you an estimate privately. But generally IVF, people worry about like, oh, it's going to be expensive, and then this whole process adds on God knows how much, and it's just going to be billionaires doing this, and then their super babies will run around like smacking us with sticks or something. And it's not really the case. More and more people are doing IVF. Obviously, it started for infertility, and people realize you can use it for making sure your kid doesn't get Tay-Sachs or whatever. Sorry, I keep relying on Tay-Sachs, but it's a fun one. Yeah, and there's a lot that can be automated here. It's a very manual process. You get these tiny little glass micro needles, and you got to like hunt sperm down and chop their tails off so they stop running, and then you pick them up and shove them in here, and you got to like turn this egg, which is like tens of microns wide, and then get just the right angle, or it gets all fucked up. But robots, AI, etc., kind of stuff. I'm sure someone here knows about AI and how it works for not killing us all, but vision stuff. But it's not really that hard to control. Other than that, this is basically all the same process as IVF. So it's IVF plus growing cells in a dish, which you can find biologists to do that. We're very cheap. And you can do that basically in the same, in the clinic, in the IVF clinic, or somewhere else, and kind of ship them around. Those require a decent amount of genome sequencing. If you look around, especially if you're getting a bulk discount, Illumina stock price is down, you can probably do 200 per genome for each of those little colonies, or do, kind of snip arrays kind of thing, and then if one looks promising, do full genome on them. And like I say, this, with a good amount of funding, 12 to 24 months, you could probably get this done. You're not paying off like amortizing R&D on this kind of stuff forever. And yeah, people already are happy to pay for IVF to do, a couple IQ points or that kind of stuff. And if it's, 100 grand on top of that or something, you'd have quite a large market. Yeah, there's economies of scale with IVF. When it's not 1% of the people, it's 5 or 10%. That'll help. So I covered the FDA stuff. There's a few options. Change the laws, which I always put on here as kind of a joke, but then it will apparently happen. So, but I'll still go through the slide. It's still fun. So the other options, or maybe that gets killed in the Senate or something. There's technically offshore, off the Greek islands for some reason, a very popular spot to do this stuff. You have your little floating clinic. There's I put Fordlandia 2 on here, but that's basically Roatan down in Honduras. You get enough infrastructure, you can get liquid nitrogen out there, it's not that bad. Ukraine obviously is, but they had basically no restrictions on this. They were a hotbed of weird IVF surrogacy stuff. I think that was the only place you could do that 3-parent baby, the mitochondrial replacement therapy, up until the war. And the UAE, fun story with JK from before. Someone got access to his emails when he was doing the designer baby stuff, and there's a clinic in the UAE that emailed him like, hey, we'd like to license your technique. So I guess they thought it was doable over there. Kingdom of Saudi Arabia, they, in their Vision 2050 plan, along with that stupid city that they have, they also put, human germline editing, and I guess people just focused on the city bit for some reason, which, is stupid, but, works for us. China, they put some laws on the books about this stuff after JK, but they have the little island down in the southwest, Hainan, where they set up weird medical stuff to do maybe. And
they also took down the firewall a few days ago there. I don't know.
Oh, sweet. Okay, yeah. I'll maybe visit sometime. And then various other like micro states. A lot of them are like tax havens or whatever, but they could do a decent sideline in this. kind of have people fly out. Like, even if this remained illegal in the US, it's, right, the illegal bit is, implanting the embryo into the uterus. So you just fly somewhere and they do that, then you fly back. You're basically good. Again, no one has tried it yet to find out. The other option that I think would be cool in cyberpunk is black clinics, just underground. You get some guy with a tattoo doing gene editing, but that would probably just be too stupid to. Yeah, we can. Yeah, what?
Like you're enlightened.
Yeah, exactly. I mean, theoretically, if there was a basement that no one knew about. So that would kind of fucked up the. But whatever, I'll just read it out. So yeah, like I touched on before, it's not just going to be the rich. Probably, the first few are going to be people who are willing to subsidize a lot of the kind of R&D work, the basis of this, but after that, it won't really be that bad. And frankly, if I could get, the state of the art in gene editing right now, or like $100 million to inherit, I would take the money every time. You can, yeah, honestly. I mean, we could do a poll or something, but that's just what I would do. And then, so there's the church quote, which kind of got cut off. We're not necessarily opposed to enhancement if everybody gets access to it simultaneously, which is not really how technology works. And I understand this is a very exciting and powerful technology, and you want to really hope that this would work this one time compared to like cell phones or whatever. You don't really need them, but that's really not how these things work. So, I love George and everything, but I think he was just, well, partially I think he was just saying this so he didn't get too many questions asked of him, but I'll just leave it at that. So hang on, let me get some water. Yeah, the ethical stuff. Again, this is probably not the right crowd to preach about this, but yeah, I might as well. So obviously, there's always a risk with this kind of stuff. With every edit, there's always a risk of something going wrong. The more edits you do, there's definitely a balance to be found there. But, as long as you're doing what is best for the child, it's, I would say, ethically justified, morally, whichever the difference is there. Children, they don't consent to being born in like a crack house. So we don't really seem to restrict that. I think this is probably better than that, and people are free to disagree, but they're wrong. And we already, face this every day. I think one of the earlier talks we discussed, deaf people used to, and this is apocryphal, but they used to do this totally, is deaf people in like 1800s or before would have a hearing child and they'd just get a screwdriver and then the kid's deaf. And yeah, deaf people are just capable enough to be assholes, but not, yeah. And we, and like I say, yeah, we discussed like, oh, we should let them do it. I know, it's a tough moral question, whether, if you're avoiding other regulations, it would suck to let people do that. And infertile people, obviously, if you're going in for IVF, especially for male infertility, which is what this allegory is based on, more than likely, I'd say, probably 50% chance if they have boys, the kid's also going to be infertile. There's a old biology joke being like, is infertility hereditary? And it's like, haha, and yeah, now it is. So, these are the kind of things, and obviously the kid can just, go out and get their own IVF in 20, 30, 40 years when they have kids, but it's a disability, and we're, happy to give it to them. It's up to the parents, frankly. So, an often cited argument is, well, why don't you just use this first to cure diseases? And there's not really any benefit there. If you're already getting embryos for this, you can probably find one that is not again, Tay-Sachs sufferer, for example. You can just, even if both parents are carriers, 75% of the embryos are going to be not actually experiencing the disease, and you can just pick one of those and you're done. It's very rare edge cases. I think beta thalassemia is the only one where both parents can have it. This is a recessive disorder, and then they are actually healthy enough to have kids, and I guess they think that they should. And that's where most of the research using this as the excuse has come from, but all this oh, well, that'd be totally cool if we did do it, and everyone agrees with that. It's like, yeah, but that's really, we don't need to. So it's for enhancement. That's basically the summary of this, and that's cool. And then people say, well, what about gene therapy? Yeah, it sucks now, and it's sucked for a long time, but maybe magically 20, 30 years from now, it'll work. And then, we'll be cured, and all these kids, we don't have to mess around with their genes. They can, consent and fill out a contract, and then they can get magical gene therapy as well, and this all becomes a moot point. And that would be nice, but gene therapy is very difficult, and your body does not like having a lot of viruses shot into it, and that's a good thing. But the problem is, you do that, maybe if you're lucky, you can do it once. It's not really hitting your entire body. It depends what you're trying to do with it. If you just want to hit the liver, basically all liver diseases will be cured in the next 5-10 years probably, because all these things just kind of end up in the liver really. And then if you're trying to deliver a foreign protein, like let's say there's some protein that dissolves arterial plaques, and we don't have one, so it's going to be a foreign protein that you're injecting, and then the body's like, oh, that's not me, so it attacks it and destroys it. Whereas if you put it in at the embryonic stage, it's already in your genome, your body thinks it's part of yourself, and you don't have to worry about that. And gene therapy continues to suck. So I know there was some, early talk about, oh, we can just inject genes into the brain and do stuff, and even if you're replacing your entire body, you're still moving the brain over, and then if your brain gets Alzheimer's, it doesn't matter if you're, got washboard abs or whatever on the bottom, you're still 110 years old up in the brain. And the brain, obviously, if you get an infection in the brain, that's bad. Every other tissue in the body, you lose 10% of it, whatever, to the virus and from the immune response. It doesn't really matter, you just regrow it. You lose 10% of your brain, you're kind of fucked. So you want to be very careful with that. It's also got a skull around it. It's got the blood-brain barrier. It's got the dura mater, all the other membranes I forget because I don't like neuro. But it's tricky, and the cells are, neurons at least, are non-mitotic, and a lot of gene therapies rely on the cells dividing to kind of dissolve the nucleus, allows stuff to sneak in there, and that does not happen with neurons. So it's tricky. And then the stem cells, right, if you just shoot some of this stuff in there, right, it hits most of the cells. And then the cells, they have a certain half-life, as it were, and then they die and get replaced by fresh cells pumped out from the cell factory, which is these stem cells. And these stem cells are very important, as you can tell from the name and the way people talk about them, but they're not just fucking floating around out there the same as every other cell. They have a niche that they're protected in, because if you lose a stem cell or a stem cell gets infected, that's much worse than any other cell getting infected. So they are protected from environmental insults. And if you don't hit those, then you got to keep doing the therapy, but then you already have the immune response, and it's just kind of, it's not great. Also, it's, expensive, and there's a reason there's only like 2 approved gene therapies, and they both cost millions of dollars. So that's why I like germline therapy. So there are a few lists of cool mutations to make. Church, as much as he likes to claim we have to wait for everyone to be ready for this, he's already kind of ready to go with his list. This is only a partial list. And there's more other private lists. I'm surprised he still has this up. It's very hard to Google it if you just kind of search this kind of stuff, but it's still up, so respect there. And most of these are going to be mutations that already exist in the population, subpopulations, and it's like, oh, these people get less cancer, or less diabetes, or their bones don't break. The ABCC11, that's a fun one. A lot of East Asians, you sweat, but you don't secrete the sebaceous glands. That's why they have dry earwax. So it doesn't get, it's pretty sweet. You just don't secrete fatty acids that bacteria break down and make smell bad. I'd get some of that, that'd be sweet. But there's a bunch of other stuff on here. Some of these are more or less developed or reliable or speculative, but I'll get into some of those as well. So yeah, where can we look? Obviously it helps if you're trying to convince.
Don't use those pictures anymore.
Inbred populations.
I'm just saying, they all like wearing hats for some reason. I don't understand it. It's some sort of common trait. But a lot of them, like the short sleep stuff, was originally discovered in, I don't know if they were, not that half, no. We split from them a while ago, yeah. Anyway, it helps convincing the general public. They go, oh, you're playing God. And it's like, well, these people playing God when they have babies? I don't know how good of an argument it is, but it's something. So like the short sleep stuff was in, I don't know if it was Amish or Mennonite or one of those fucking, Pennsylvania types. Ashkenazi, we mostly have negative ones of these, but we also have some good stuff in there from the old Shtetl inbreeding. But there's plenty of other stuff. If you just look at human evolution in general, the paper when they first sequenced the chimpanzee and the human genomes, the organ that had the most number of changes in it was not actually the brain. Does anyone know which organ had the most? I don't even know if that's closer. It's kind of a trick question because it's not usually considered an organ, but it's the immune system. Survival of the fit does not mean you're like slaying your enemies and climbing up a mountain and like harvesting grain more effectively. It means when cholera comes through your village for the 4th time this month, you lived and some other people didn't, and that's really the competitive advantage. So we have massively overdeveloped immune systems, various other disorders that are help you to not shit yourself to death, like cystic fibrosis, which is good. The obesity stuff, if you get famines after famines, same with muscle atrophy, we don't really have these problems anymore, and if they do come back enough to be an issue, we're kind of fucked anyway, so we can kind of revert a bit back to the mean, the not quite, paleo level, because that's kind of a scam as well, but a lot of these adaptations were made because they're expedient, not because they're, good for longevity or healthy living or anything like that. It's, again, a miracle we survived it all, especially through civilization. So there's some options there. There's some good ones. There's also, animal studies are good. I would like to draw the line between physical trait kind of stuff. There's some stuff like the Pepsi K mice, the mighty mice. They can you just put them on a treadmill and they just don't stop really. They eat a lot of food, but again, if we're running out of food, we're kind of fucked anyway, so that's not really a big deal. Cancer, the saying we've cured cancer 100 times in mice. It's sort of true, but there's probably something there. Quadruple muscling, you can turn that off and on, so it's not like you're just born and you're like doing 25-pound dumbbells or something, But it's very simple to do that one. It just hasn't been done in humans. And the life extension, obviously mice don't live nearly 50 years, so that would be just in mouse years, that would be a very long time. And that's the kind of thing you can at least look at. You can put it into monkeys, you can put it into other mammals and just kind of see how it does. The brain is trickier, at least for these things. Obviously there are, SNPs that we can look at in other human populations, but just because a mouse is good at finding cheese or whatever, I wouldn't necessarily go, oh, this is, the key. Like, we got the brains, not the mice, so I don't care how smart you can make a mouse. That's not really going to be that relevant for us. And then the fun one, which is probably going to lose a lot of people if I haven't already. So the kind of the promise of adult gene therapy is, I'm just calling it embedded pseudovirus, but the idea is you kind of design a synthetic virus, which is just, there's like the proteins that wrap around the DNA, and then there's one that kind of forms the little blob membrane, and then there's the outer bit, like the COVID spike protein that interacts with receptors on cells, and you make one of these, no relation to any existing viruses out there. You can just kind of design one of these, and then you stick a copy of this genome into the genome of the kid along with the other edits. And the cool thing about the thymus, so your immune system is trained not to attack you, and usually it works. The way it does that is the thymus is around, and then it eventually kind of shrivels up and dies. It's very sad. But what it does is it kind of conga lines all of your immune cells through it, and if any of them would attack your self proteins, they are told to kill themselves very politely, and that's how you prevent autoimmune disorders. And you can just put this kind of virus genome in the genome of the human, and it's not really expressed anywhere else. You're not just making little viruses in your day-to-day life, but it just means the immune system will not react to it. I won't go into promiscuous gene expression, but it's pretty cool. So you just leave this in there, and then you wait, and the kid grows up, and you go, oh damn, here's a really cool SNP edit that we could do, and then the magic happens. And this is not the best diagram. So here's the the virus with the genome, and then there's the human who also has the genome, not to scale. So you decide what edit it's going to make. You could have like the guide RNA or whatever, and you grow a little bit of this virus in a flask, and then you inject it. And here's your average everyday cell from this early modified person, and the virus comes in and attaches. There's your little, oh yes, there's also a synthetic receptor that you add that doesn't do anything except bind to this virus, and that's just kind of a little bit is on all your cells. So the virus comes in, hits that, injects its stuff like viruses do, and does the edits, and then it kind of suppresses that receptor. So the cell, this is a light blue force field. The cell can't get reinfected. It makes a few more viruses, and then it kind of stops, and then the kind of process cascades. One of the major problems with gene therapy is adult gene therapy is you're using non-replicating viruses, because that would be bad for reasons I can go into later, but and that means you have to make a shit ton of virus and inject it all at once and just hope it does what it needs to. This way, it's more mimicking the natural viral process. It kind of spreads around, it goes from cell to cell. Oftentimes it doesn't even have to bud off from the cell, it just kind of passes next to it like HIV does. And then eventually, skipping over many of these replication cycles, all of your cells have the modification, and there's a bit of virus floating around, and then you can do like TET response element. You can basically reset this whole thing so the cells have the receptor again, and then you can do this again if you want to. And this, would need a bit of work, but less than you'd think. And this is kind of the future proofing, right? People don't want to have kids now because they're worried they're not going to have, the right selection stuff or the early designer baby stuff. But no matter what, you're going to miss out on something, and this allows you to kind of somewhat future-proof your kids, which It's probably a new phrase, but I like it.
Excuse me.
Yes.
So the idea here, I'm just a little bit confused by that. You're basically editing you're giving some kind of virus to the embryo that then makes it able to be edited later much more easily?
Yeah, you just put in like a little chunk of DNA that encodes the virus, but the promoters don't really express anywhere in the body, so it's just kind of silently waiting in there. Like half of our genome-ish is endogenous retroviruses that kind of got in there and then they kind of jump around a bit and replicate and whatever, but that's most of the junk DNA. It would be most similar to that, yes, but it wouldn't be doing anything really. It's just the code for it, but not the actual virus being produced. And there's a bunch of other edits. People always ask me what my favorite edits are, but whatever. That one was mine, even though it's a bit more involved than the usual kind of stuff. So I'm sure everyone's kind of seen these, especially the one on the right kind of thing get cut off there. But this was over in China on, for some reason, if you're not Christian, you don't really care as much about the ethical implications about this kind of stuff. China, fairly positive. Over here is a bunch of different countries. I had to, it's a good thing I did zoom in on this. India, I'm not going to attribute this to the caste system or get involved in that debate, but they are super cool with like making their babies smarter. Way more than anywhere else. Not that they pulled all the countries here, and these things are malleable, but something to note. Yeah, of course they still have laws in the book against it, but yeah, yes. Oh, yes.
Yeah, I'm sorry if this is an ignorant question. I'm not a biologist. How does this work for epigenetic phenomenon or emergent phenomena? Even IQ considered emergent in some respects?
Yeah, we're not really touching that. I mean, a lot of epigenetics is bullshit anyway, but mostly it just kind of sorts itself out. Epigenetics is determined mostly by genetics. So
Most probes are reverse causal still.
yeah, it's
Most probes don't actually indicate like a thing that's affected by genetics. They think that something is leading to the marker, the methylation or whatever.
Well,
we,
let's say
we can explain later. I wouldn't worry too much. The main thing is the kind of imprinting at that early stage, making sure everything's where it should be at that point. But yeah, going from one cell to 10 trillion cells is a lot of epigenetic stuff that's constantly changing, and it's most of it's not set in stone. Any of the histone methylation stuff doesn't really matter. Really, it's only DNA methylation. But yeah, it'd be the same as a natural baby in that sense. It's you just kind of trust epigenetics to sort itself out, really.
I wrote a really good explainer that Luigi Mangione really enjoyed.
Oh, good. Yeah. Maybe we can bring him to Austin when he gets out. So, anyways, the general gist of this is, yeah, like since the 40s really, but especially since like the 70s when they started the whole war on cancer thing, it's like, oh, here's a cool gene that helps you out and would substantially reduce the risk of cancer, but actually using that to help people, you can try to find some small molecule that maybe targets it and doesn't have horrible side effects and isn't too toxic, or as a gene, but gene therapy is basically impractical. And it's just like we have quite a wealth of knowledge here that we could apply, and this is the best way to do it, and we should do it. And I make the argument that the longer we delay doing this, for various bioethics concerns, is another generation of babies that are born without, even the smallest of benefits. Yeah, and the first generation stuff, it's not going to be like fucking crazy, and they're not going to be jumping off buildings. I mean, to like between buildings, they're not just going to kill themselves. I need to work on that metaphor, but you know what I mean. Yeah, like the longer we wait, and there's a lot of people who are like, oh, like I mentioned, what if adult gene therapy gets good? And it's like, yeah, what if it doesn't? And then, we're just dooming them to our same fate. So I can ramble on more about the ethical arguments, but I'd prefer to be drunk before I do that. So we can sort that out. How are we looking on time? Not bad, okay. Yeah, pretty much, we can do Q&A, yeah. Who's got, I survived. Yeah, okay, yeah, Steve.
I think you mentioned a 12 to 24 month timeline if well funded. What's the most challenging step that has to be accomplished?
Challenging, you mean in terms of like amount of time or like technical?
There's some chance we might not figure it out, or.
Yeah, so like I was saying with the Serrano lab research and Sergey's SuperSox stuff, so the Serrano lab, like I say, they did a few papers on it, and then they fucked off. And then Sergey, I asked him about the imprinting. He's like, oh, I want to do that. Maybe he's doing it right now. Maybe that's why he's attending virtually. We just need to kind of test that, and it takes time to wait for the cells and see how they grow, do all the epigenetic sequencing, that kind of stuff. From there, I mean, getting eggs and embryos is probably annoying. We kind of need both. You can just start with regular embryos, but especially for like nuclear transfer, you want a bunch of eggs. So if we have any potential donors, you'll probably be compensated. I don't know if that's legal either, but it should be. And then, a lot of sequencing, just, kind of stress testing it. You want to do, like, supposedly, I guess, Jay's gone, but I was going to say, he's claiming, you don't even need to get approval for this if they, strike this from the appropriations bill. That seems suspicious to me. I'm sure the FDA will still stick their fingers in it. So in terms of getting this like GMP, that could be a very long time. Getting it good enough that I would do it, there's probably a lower but very reasonable bar there. Just doing it hundreds, thousands of embryos really, and making sure it works reliably enough. That's the main kind of stuff. Finding people willing to work on this and not torpedo their careers is definitely a concern. Most of the people are academics. So.
When you say doing, I forgot the number, hundreds or thousands of embryos, would those be monkeys, or you're saying humans?
What are you, a cop? Yes, of course, monkeys. I know some people want to do monkeys first, but I mean, even just like human embryonic stem cell culture is much more developed than like marmoset stem cell culture. So if it becomes legal, then there's no reason not to just do humans first. So, but if we have to, we can get a little breeding colony together of monkeys, mind you, and do it that way as well if you want. But I mean, up until the point you're implanting them, we're not, generating fucked up human babies. So maybe on the side, depends how much money we get, we can have some monkeys. Basically.
Great.
But and Merrick, I'll go front to back. Sorry. Yes.
I think the monkeys are definitely going to be necessary to show that it's safe. I also had a question. How do you know which edits to make? I mean, besides like George Torch's short list, which gets you a few traits, right? Like we heard from Steve about difficulties identifying causal variants.
Yeah, I wouldn't go with the usual SNPs. I mean, I don't know how many of those are actually causal and not correlative. Like I say, when you go out of the ethnic group, the linkage disequilibrium kind of stuff. I would look more for like those rare, like basically single point mutation, like monogenic kind of stuff where ideally, it changes the protein in a way that we can understand. But some of it, like the quadruple muscling, is like, you just express dominant negative activin 2B receptor in the muscles, and it does this in mice. So it depends on the target. I wouldn't say that's not really my department, but I haven't, I'm sure we'll get plenty of options. You talk to a biologist, and everyone has their kind of favorite gene mutation to make that's fairly well studied, and we can collect a list of those, I think. But
I think Chase, I don't know if is Chase here. I
Yes,
think you might say something like, even though you're not sure it's a causal, you just do it anyway, and then like overall, like your overall effect size is a third of what you thought it was going to be or something.
Again, you have to balance it with the risk. There's always the risk of the off targets with CRISPR, and even if you're doing the sequencing, if you get some sort of cascade where you're chasing your own errors, I'm sure there's a balance to be made, and that'll be part of it as well as finding out how much multiplex kind of editing we can do. But yes.
Well, I mean, why wouldn't you just sort of lower the risk of terrible things happening by just working on superficial things to see if they work? Like dumb things, basically, that's not going to have much of an impact anyway, just to see if...
Work on, I mean, we could, sure, I guess, that's up to, I mean, we can just, like when we're doing the testing stuff, it'll just be, like, here's a random site and change it. Most of the testing we're going to be doing is not embryos or embryonic stem cells that'll be implanted. It's just cells in a dish. So.
It just removes the ethical consideration on it.
Oh, just giving people like graffiti upgrades to their genomes, like stick my name in there.
Just to see if it works.
It's kind of a dick move to do to them. Like, we could have given you immunity to cancer, but just sign my name and.
I think the issue is like you're taking some other risk in that the baby is not the embryo is not a normal embryo because you made it through these stem cells. So there's some intrinsic risk of that being bad. So if you're going to subject the kid to that, why not give him some good stuff?
You want some benefits. Yes, the whole process, while we think we can make it very good and all the epigenetics will be okay, it's still a risk even if you're not changing anything. And we can do the testing, but at a certain point, the first kid's going to have to be born with it, and we want to give them something for their trouble, really. But it's up for debate, really. Yes?
Okay, thank you. This is going to sound very
I'm barely a scientist, don't worry.
But when it comes to things that are popular, that we already know from biobank, well, knowledge that are innocuous when it comes to physical health, wouldn't like the most lucrative traits to edit be cosmetic ones? Like when it comes to like lighter skin, that's very popular, like worldwide.
Yep.
And so is height or males.
Yeah, height, eye color, hair color, skin color.
Like forget like positive editing for like super like muscles or something like that. Like when it comes to like the market, like when that where that gene therapy that
Well,
basically be where
Muscles are useful. You can lift stuff. Yeah, and honestly, from what I hear from muscle people, it's not really for attracting mates so much as like the boys kind of thing. So, but it has its own perks, really. I'm sure someone more jacked than me can weigh in on it, but it's just generally, yeah, it's good for health as well, having muscles. There's a lot of very strong correlations there. Again, like I tend to side with Jay on this, that it's not our decision to like restrict it, that you can't try and make your kid look 5% more like Brad Pitt or something. Yeah, it depends on the people. Most of the first round of people doing this are probably not really going to care about that as long as the kid is smart or just healthy in general. But I don't know.
I think some places would like to fit into like the dominant culture.
Yeah, probably, especially the skin stuff. There's people who've gotten bleached their skin or whatever. East Asia, I think as well. Yes, is a major problem there. Yeah, I don't know. People are going to just do some weird shit, but we just roll with it. I don't know. I'm not the bioethicist, and I hate them. So.
If people buy products that cause them to get cancer, then like, yeah.
Yeah, exactly. And again, there is always a risk. Like, if you're going to go through the effort of like changing the eye color, I've never noticed what someone's eye color is. I guess some people do, but is it really worth it when you could have, changed a couple more intelligent snips or something instead? It's who the fuck knows. Yes, Anatoly.
What's the source of the map?
What map? Oh, that was from some paper. I put this thing together like 4 years ago, so I don't know if it's still up to date. Like, I want to see if South Africa is still, or if they were originally red.
As of last year.
Yeah, because
Regarding the polls, I
oh,
do have
oh, it's gray.
Yeah. Because, what do these stars represent?
That's where. So I did. There's America, where we can change the laws. There's Roatan. There's Ukraine. There's Gulf states. Hainan.
The stuff you've identified.
Yes, yes.
Okay,
Sorry, it's,
and yeah, just one more thing about the opinion polls.
Yeah.
One thing I noticed about them in general is that the Asian emerging markets, they tend to be more technophile in general, so not just on biology, but also on AI in particular. So,
Good,
that
yeah.
might be just sort of some general factor like that.
yeah, I think they. There's a lot more emphasis on like general like being smart, academic performance kind of stuff there, and like I said, they don't have the Judeo-Christian, although I guess it's just Christian if the rabbis are cool with this, concerns about this, which are not really founded on anything themselves. Funnily, Italy, super Catholic country, is yellow on this, but that probably means basically the same as red. And I think Belgium, yeah. But, yes.
Coach, would it go, yeah.
What?
Could I photograph this slide in particular?
Yeah, sure.
Black clinics.
Oh, black legs.
Yeah, so it would be so cool though, right? Oh, yes.
Sorry if I'm being slow on the uptake here, but I just want to make sure I'm understanding the embedded pseudovirus thing. So,
Yep.
like, the idea is this pseudovirus doesn't itself do anything, but
No.
it's given to the embryo.
Yes.
And then the idea is we want to make all these changes for the adult.
Yep.
And then you give the adult the gene therapy, and then it works with all these problems that it normally has. But like, why wouldn't it have all these problems? Like, it still has to reach every cell and all this stuff.
Yes, I can go back to that slide. I know I was considering not putting it in there because it takes me a while even explain to biologists on this stuff, but I promise you it's cool. So the sequence that you're putting into the baby's genome here, it doesn't do anything. It's just there to tell the immune system that it's cool if you see some of this, don't go apeshit on it. And then the virus, it kind of slowly spreads cell to cell, or some of it gets into the bloodstream, and viruses want to stick to their matching receptor, and we have this kind of artificial lock and key thing we could do very easily. And virus sticks to a cell, injects its genome in there, and that floats around and does what it normally does, either editing or making more viruses. And then it just kind of slowly cycles through. This is, it's still quite a bit different from regular gene therapy. Like I say, with that, you're just injecting a massive bolus of virus, and you have them on heavy immunosuppressants for like months before and after, and that's where most of the problems come from. It's just trust me, it would probably work. basically. Sorry, if I had like a whiteboard, maybe we could do a bit better with it. Yeah.
It's like having a backdoor to your body. Like,
Yes,
with
basically.
all the problems and
Yeah, it helps a lot. It's not going to be like super amazing perfect, but this I think is a nice early idea for how to go about it. Yes.
How long is like the time between when you express like the sRNA or whatever for the receptor to like when it actually gets knocked down? Like how long do these like receptors last in the membrane?
I mean, you could make it so that they're very short-lived and they're just constantly being popped out there, and as soon as you knock down the RNA, within like an hour, they're basically gone. So it might get reinfected, but the cell, might have 5 viruses slam into it instead of 1. And with this kind of stuff, you're already encoding. For example, you could already have like Cas9, the CRISPR protein, already in these genomes as well, because you can get immune reactions to the CRISPR protein, and you can get rid of that, which by itself would be pretty cool. This is just adding the whole delivery mechanism on alongside it. So yeah, there's some tweaking to be done there. Definitely, we can test it out in a dish or mice or whatever and mess around with that. This is not going to be in the first generation, sadly. So they are not getting future-proofed, which is doubly ironic for the first few of these kids. But within a decade, I think this would be a good thing to do. Yeah, but we'll see. Yes.
What about someone adversarially designing a virus to hurt you? If you know there is this like nice receptor.
You're only like the 5th person to ask me that. So there's a few options there. These are all kind of synthetic, like the receptor, like the spike protein and the receptor it's like lock and key. You can design a binder, like 2 proteins that stick together from nothing, basically using AI kind of stuff. So you can give everyone their own one if you want to, although that's probably annoying logistically to make everyone their custom gene therapy when you gotta like look this up. You can do a bunch of those. You can also have, so you get to this stage where all the cells have been infected, you get your first gene therapy, and you basically just don't reset the receptor so that they are functionally immune to this virus until you give them the doxycycline and reset it. So there's a number of methods. If someone really wants to kill you, they'll just shoot you in the face. Just all the engineered bioweapon stuff is...
For what it's worth, I think this was one of the potential failure scenarios of the AI 2027 timeline, which was like a latent...
I'm not really on less wrong, so you'll have to fill me in on what that means.
Like a latent virus that infiltrates the entire populace, and then later on is triggered, right? So,
yeah, I mean, I'd be more worried about like prions. Frankly, you could have some doomsday scenarios there, but I don't want to scare people. Yes?
Yeah. There's so many easier ways to kill people.
Yeah,
right?
I mean, if Luigi showed us anything, yeah.
So I just wanted to clarify. So I'm pretty sure I get it. So the idea is that if you are able to put the genetic material into the embryo at the beginning so that the body is essentially like, oh, this is one of us,
Yep.
then the body doesn't attack, and then later you can extract, or like build the virus, right? That is that exact genetic sequence which the body won't reject, and you can use it for whatever you want.
Yeah,
So
exactly.
you're just Trojan horsing the body.
Yeah, basically, yeah.
Yeah, okay.
It's pretty cool. Yes, in the back.
Yes. So in this embedded pseudovirus example, I guess it solves the immune system problem, but how does the administered gene therapy end up crossing like the blood-brain barrier, for example?
Yeah, the brain's probably tough. You might have to do a separate injection just into the brain, and then it'll, it's passing through not just neurons, because they're going to be kind of annoying, but all the microglia and astrocytes that are floating around there, normally dividing. They will provide a fertile host for the virus to kind of do its thing. So yeah, you probably still need to shove a needle into your brain. We can get really tricky with it, but that's, probably the easiest way. Yeah.
Or
There are ways.
The epithelial cells could express the receptor, right?
Yeah, there's probably, it's like theoretically the blood-brain barrier is made up of cells, and they are, they take it in on the blood side, and then a few of these viruses are getting excreted on the brain side of it, and you can get some pass-through that way. Yeah, there's a number of ways there. But yes, the brain will be definitely an important one, so we want to make sure we get that. This is the first time I've done this talk where I put the embedded pseudovirus in. I'm glad everyone likes it, but yeah, cool. All right, is that it for questions? Any last minute? You can just find me. I'll be around. So, yeah. Thank you.